Welcome to this microbiology video where we're going to talk about how to do an acid-fast stain. Now with acid-fast stains, what you need to understand is that this is a special type of gram-positive bacteria that have certain molecules known as mycolic acids incorporated within the cell wall that are extremely hydrophobic, not allowing many molecules through the cell wall. This makes them very difficult to treat and very difficult to stain as well, especially when you do a gram stain. Even though they are technically gram-positive based on their cell wall structure, they do not soak up much of the crystal violet and they end up showing very faint purple if dyed at all. So this differential stain is designed to circumvent that mycolic acid so that we can actually visualize these specific types of bacteria under the microscope. So what we're going to be doing is something that is very similar to the endospore stain procedure. A lot of it is the same, so we're simply going to talk through it here for a moment and then show you how to actually perform the stain. We've already prepared everything up to the point of performing the stain. But to get to that stage, remember you need a slide, label it correctly. You're going to have slants that you're drawing your cultures from, and because these are solid cultures, remember you need to prepare your smears using some sterile deionized water. When you do that, make sure that you're adding the water to your slide with your inoculation loop, sterile, then collect the solid culture with a sterilized stab, and then spread that around with the inoculation loop. It is critical, especially with some of these species such as the microbacteria. The reason for that is they are extremely sticky, and if you do not take the time to spread them around, you're only going to visualize a giant clump and you're not going to have anything worthwhile imaging. So make sure you spread them around. Okay? Once you get your smear dried and heat fixed, then we get to the actual stain process, which we're going to show you here in this video. So the first step is get your slide in your glass bowl with wet paper towel and water underneath the slide so that there's even transfer of heat across the slide. And then we're going to use the Ziehl-Neeson dye in the fume hood. The reason for this is the Ziehl-Neeson dye is not a very fun dye to work with. It's a nasty dye, so we want to limit our exposure to it. So add the dye here. Make sure you cover where you have your smears. And then just like with the endospore stain, we're going to microwave for 30 seconds. So why are we microwaving the Ziehl-Neeson? That mycolic acid, as I mentioned, is extremely hydrophobic. Not many molecules are allowed past the mycolic acid for that reason. The heat is acting on the mycolic acid to loosen it up just enough that this Ziehl-Neeson dye can get through the mycolic acid and into beneath the cell wall. When we stop applying heat, the mycolic acid is no longer excited and warmed up and is no longer allowing things through, and now the dye that made it through is trapped underneath the cell wall. So again, be very careful when you pull this out of the microwave. It shouldn't be hot enough to burn you, but be careful. We do have hot glass holders there, and also be very careful with your slide. It may also be. So once you get your slide here, you're going to take your acid alcohol wash. Now why are we doing an acid alcohol wash? If you use your normal ethyl alcohol wash, it's not going to work. And this acid alcohol you do for 8 to 12 seconds, preferring the 12 seconds. There we go. And then immediately follow up with water, again, so we don't over decolorize. So, whenever you're doing this, make sure you do not accidentally ethanol decolorize wash. Use your acid alcohol. We will have these appropriately labeled, so make sure you use that for your decolorization step when you're doing an acid-fast stain. Once we've rinsed the acid alcohol off with water, we can then come in with our counter stain, methylene blue. Again, simply add it to where you know you put your smears. Cover them completely. Let them sit for a minute. Okay? Now, with this, what is the methylene blue doing? This is your counter stain. This is your second stain that you're coming in with. If everything went according to plan and you have a positive acid-fast cell, what you're going to notice is that red soaked in through the mycolic acid when we heated it up and got trapped underneath it. Now, after we decolorized, any excess red got washed off the cells. Okay? So now, unless it's an acid-fast stainable cell, that red is not going to be present. The blue is simply coming in to stain any cells that do not now have stain on them after they've all been decolorized. Okay? So, once our minute is up, we will wash this off with water. There we go. Here's our water. Rinse it off just as before. Be very careful not to spray directly onto your smears so as to not knock the bacteria off with forceful spray. And once it runs clean, dab it dry with bibulous paper. Again, be very careful not to rub the bibulous paper. Simply pat dry. And you now have an acid-fast stained slide to look at under the microscope. Now, once you have your slide stained, great. How are you going to get rid of these? These stains that we use with both endospore and with acid-fast stains, they are very environmentally unfriendly. And so what we're going to do is we're going to have you keep the wash-off in your dye trays, and the TAs will come around and collect that for us to dispose of all together at once. So do not pour these down the sink.